In this video, we are going to learn about diffusion, osmosis, ion channels, active transport, membrane pumps, and their difference. So, transport across cell membrane. Cell membrane is a phospholipid bilayer that regulates the entry and exit of molecules. Two types of transport is there: active and passive transport. Passive transport: in this biological process, energy is not required for transporting the molecules as the biochemicals move from a region of higher concentration to a region of lower concentration. All particles which are easily soluble are transported through passive transport. This process is carried out to maintain the balance and the equilibrium level in a cell. All the waste molecules, including water and carbon dioxide, is separated and moved out of cell using passive transport. Nutrients like oxygen that are functional for cell are diffused in this process. Osmosis, diffusion, facilitated diffusion are some of the examples of passive transport. Active transport: it is the movement of molecules or ions across a cell membrane from a region of lower concentration to a region of higher concentration against the concentration gradient. Active transport requires cellular energy to achieve this movement. Active transport is only commonly accomplished by the transport protein that undergoes a change in shape when it binds with the cell's fuel, a molecule called adenosine triphosphate or ATP. Active transport is mainly two types: primary active transport and secondary active transport. So, difference between active and passive transport, we will be dealing with them separately. First, let's learn about active transport. Transport of ions and molecules is from their low to high concentration. Cellular energy is utilized for transport. Transportation is highly selective. Pumps are involved in active transport. There is a fast process. This is a fast process. Active transport helps in the transport of large cells, proteins, ions, complex sugars, etc. Active transport is influenced by temperature. Transportation is only in one direction, like potassium sodium pump, sodium calcium exchanger, etc. Then passive transport is also there. Transport of ions and molecules is from high to low concentration, does not utilize cellular energy for transport. Transportation is pretty selective. Pumps are not involved. Only carrier proteins and channels proteins are involved in facilitated diffusion. This is a slow process. Passive transport helps in transporting soluble molecules such as carbon dioxide, oxygen, water, etc. Passive transport is not influenced by temperature. So when we compare this to, in case of active transport, transport of ions and molecules is from their low to high concentration. So this requires energy. So transportation is highly selective. Pumps are involved in active transport, fast process. Active transport helps in the transport of large cells, proteins, ions, complex sugars, etc. Active transport is influenced by temperature too. And sodium potassium pump, sodium calcium exchange, etc. are the examples. But in case of passive transport, transport of ions and molecules is from high to lower concentration, does not utilize cellular energy for transport. Transportation is partly selective. Pumps are not involved. Other carrier proteins and channel proteins are involved in facilitated diffusion. This is a slow process. Passive transport helps in transporting soluble molecules such as carbon dioxide, oxygen, water, etc. Passive transport is not influenced by temperature, but transportation is bidirectional. Next part is the diffusion. Diffusion is a spontaneous process in which a substance moves from a region of high concentration to a region of low concentration. Molecules said to move down a concentration gradient. It is a passive process, which means that no energy is needed. Molecules diffuse until they are evenly spaced apart and equilibrium is reached. Diffusion is a direct result of Brownian motion. Diffusion can be divided into two main types, namely simple diffusion and facilitated diffusion. Diffusion is a direct result of Brownian movement. Temperature, area of interaction, size of the particle, concentration gradient are all factors that affect the process of diffusion. Diffusion is a very important process occurring in all living beings. All the living organisms exhibit one or other form of diffusion, allowing the movement of the molecules during various metabolic or cellular processes. First one is the simple diffusion. Simple diffusion is a process by which molecules, atoms or ions diffuse through a semi-permeable membrane down their concentration gradient without the assistance of transporter proteins. A semi-permeable membrane is a membrane that allows certain molecules, atoms, or ions to while blocking others. Small uncharged atoms or molecules are able to diffuse through the cell membrane via simple diffusion. Examples of simple diffusion occurs with gas molecules diffusing across a cell membrane. Carbon dioxide and oxygen molecules are small and nonpolar. They cross the cell membrane by simple diffusion. In case of passive movement, non-mediated passing is happening, but in active transport, transporter mediated passage is open. O2 just diffuses into the cell. Na travels with the help of carrier proteins. Four basic mechanisms by which solute molecules move across membrane. Simple diffusion through a bilayer, which always proceeds from high to low concentration. Simple diffusion through aqueous channel formed within an integral membrane protein. Facilitated diffusion in which solute molecules bind specifically to a membrane protein carrier. Active transport by means of a protein transporter within a specific binding site that undergoes a change in affinity. Movement occurs against a concentration gradient. Diffusion of substances through membranes. The substances must be present at higher concentration on one side of the membrane than on the other, and the membrane must be permeable to the substrate. Simple diffusion leads us to consider the polarity of a solute. One simple measure of polarity of the substance is its partition coefficient. Partition coefficient is the ratio of its solubility in a nonpolar solvent to that in water under conditions where the nonpolar solvent and water are mixed together. After factor determining the rate of penetration of a compound through a membrane is its size. If two molecules have equivalent partition coefficients, the smaller molecules tend to penetrate the lipid bilayer of membrane more rapidly than the larger one. Very small uncharged molecules penetrate very rapidly through cellular membranes. Membranes are highly permeable to small inorganic molecules such as water, oxygen, carbon dioxide. Larger polar molecules such as sugars, amino acids, and phosphorylated intermediates exhibit poor membrane permeability. Polar molecules such as sugars, amino acids, and phosphorylated intermediates. So next learn about facilitated diffusion. Facilitated diffusion is the transport of substances across a biological membrane from an area of high concentration to an area of low concentration with the help of a transport molecule. Concentrations move along the direction of their concentration gradient. Chemical energy is not directly required. Substances soluble in lipids diffuse through the membrane faster. Substances are hydrophilic and large molecules are difficult to pass through the membrane. Facilitated diffusion is the transport of substances across a biological membrane from one area of higher concentration to other area of lower concentration with the help of a transport molecule. Substances move along the direction of their concentration gradient. Chemical energy is not directly required. Substances soluble in lipids diffuse through the membrane faster. Substances are hydrophilic and large molecules are difficult to pass through the membrane. Examples are glucose and amino acid transport, gas transport, and ion transport. Facilitated transport. Transport protein will be there. So transported molecule enters into the inner side of the cell. Facilitated diffusion doesn't need energy. Chemical energy is not required. Selective permeability. Integral membrane proteins allow the cell to be selective about what passes through the membrane. Channel protein. They are the integral proteins present in the biological membrane that allow the transport of molecules across the membrane by forming a channel. Many channels are very selective. Carrier protein. They carry molecules across the membrane. These proteins bind to specific regions in the molecule, causing conformational changes, and then move the bound molecule to the interior of the cell depending on the concentration gradient. Glucose transport, an example of facilitated diffusion. Glucose is a body's primary source of direct energy, and most mammalian cells contain a membrane protein that facilitates the diffusion from the bloodstream into the cell. A gradient favoring the continued diffusion of glucose into the cell is maintained by phosphorylating the sugar after it enters the cytoplasm, thus lowering the intracellular glucose concentration. Humans have at least 5 related proteins that act as facilitated glucose transporters. These isoforms are termed GLUT1, GLUT2, GLUT1 to GLUT5. Extracellular space is there, transporter will be there, cytosol is there. Glucose hexagon binds to a site on the transporter that faces the cell exterior. Glucose binding triggers a conformational change that exposes the glucose binding site to the cell interior. Glucose dissociates from the transporter. The transporter converts to its original conformation. So glucose dissociates from the transporter. The transporter reverts to its original conformation. Insulin is a hormone produced by endocrine cells of the pancreas and play a key role in maintaining proper blood sugar levels. An increase in blood glucose level triggers the secretion of insulin, which stimulates the uptake of glucose into various target cells. Insulin responsive cells share a common isoform of the facilitative glucose transporter, specifically GLUT4. When insulin levels are low, these cells contain few glucose transporters on their plasma membrane. Rising insulin levels act on target cells to stimulate the diffusion of the cytoplasmic vesicles to the plasma membrane, which move transporters to the cell surface when they can bring glucose to the cell. Raising insulin levels act on target cells to stimulate the fusion of the cytoplasmic vesicles to the plasma membrane, which moves transporters to the cell surface where they can bring glucose into the cell. So in the absence of insulin, glucose cannot enter the cell. So glucose is there, insulin receptor is there, then secretory vesicle is there, resting skeletal muscle of or adipose shell. Insulin signals the cell to insert GLUT4 transporters into the membrane, allowing glucose to enter the cell. So insulin binds to receptor, signal transduction cascade, exocytosis, glucose enters the cell. Difference between diffusion and facilitated diffusion. The difference between simple diffusion and facilitated diffusion. Simple diffusion is a passive transport and does not require energy. The speed of simple diffusion is low. This is not inhibited by an inhibitor molecule. If simple diffusion, the movement of particles takes place with the direction of the concentration gradient. Simple diffusion is involved in the passage of small molecules along the non-polar molecule. In case of facilitated diffusion, it is a passive transport mechanism that doesn't require energy, but some facilitated diffusion process can be active. The speed of the facilitated diffusion is high. This can be inhibited by specific inhibitor molecule. In facilitated diffusion, the movement of molecules can take place in both ways along the direction and opposite of the concentration gradient. Facilitated diffusion is commonly involved in the movement of as large as polar molecules across the biological membrane. Now we are going to learn about the next process, that is nothing else but the osmosis. Osmosis is the spontaneous net movement of solvent molecules across the selectively permeable membrane from a region of higher water potential to a region of low water potential. Next, osmotic gradient. Difference in concentration between solutions on either side of semi-permeable membrane called osmotic gradient. So membrane is there outside of the cell, inside of the cell, high water concentration, low water concentration, direction of movement. Types of osmosis. There are many types. Osmosis has two types, endosmosis, exosmosis. When a substance is placed in a hypotonic solution, the solvent molecules move inside the cell and the cell becomes turgid or undergoes deplasmolysis. This is known as endosmosis. Next is the exosmosis. When a substance is placed in a hypertonic solution, the solvent molecules move outside of the cell. Hypertonic solution. The solvent molecule move outside of the cell and cell become flaccid or undergoes plasmolysis. This is known as exosmosis. When we just look at the pictures, here you can see a blood cell. Red blood cells kept in hypertonic solution, hyper concentrated solution. What happens? H2O leaves, so cell got shriveled. Next, plasma cells kept in hypertonic solution. Plasma membrane draws away from the cell wall. H2O leaves. When the same cell, plasma membrane drawn away from the cell wall, this plasmalized cell kept in hypotonic solution, H2O enters the cell, turgid or swollen cell created. Next, hypertonic solution. The concentration of solute is higher outside the cell than inside the cell. Have more water inside the cell, so water moves out of the cell. A cell placed in a hypertonic solution rapidly loses water by osmosis and shrinks. 20% solute concentration, about 80% water. Water moves out of the cell. 40% solute concentration, 60% water. When we just look at the hypertonic solution, we can see that a solution that has a lower solute concentration than other solution, water particles will move to the water cell, causing the cell to expand. 20% solute concentration, 80% water. Water moves into the cell. 10% solute concentration, 90% water. Isotonic solution, a solution that has the same solute concentration as another solution. There is no net movement of water particles and the overall concentration of both sides of the cell membrane remains constant. 20% solute concentration, 80% water. 20% solute concentration, 80% water. So water moves in and out of the cell. The effects of differences in the concentration of solutes on opposite sides of the plasma membrane. A cell placed in a hypotonic solution swells because having a net gain of water by osmosis. A cell placed in a hypertonic solution shrinks because of a net loss of water by osmosis. A cell placed in a isotonic solution maintains a constant volume because of the inward flux of water is equal to the outward flux. Net water gain, cell swells in case of hypotonic solution. Net water loss, cell shrinks in case of hypertonic solution, and no net loss of or gain in case of isotonic solution. The effects of osmosis on a plant cell. Aquatic plants living in fresh water are surrounded by a hypotonic environment. Water therefore tend to flow into the cell, creating target pressure. If the plant is placed in a hypertonic solution such as seawater, the cell loses water and the plasma membrane pulls away from the cell. Normal target pressure, but in case of hypertonic solution, no target pressure. Significance of osmosis. Osmosis influences the transport of nutrients and the release of metabolic waste products. It is responsible for the absorption of water from the soil and conducting it to the upper parts of the It is responsible for the absorption of water from the soil and conducts it to the upper parts of the plant through the xylem. It stabilizes the internal environment of a living organism by maintaining the balance between water and intracellular fluid levels. Osmosis influences the transport of nutrients and release of metabolic waste products. It is responsible for the absorption of water from the soil and conducting it to the upper parts of the plant through the xylem. It stabilizes the internal environment of a living organism by maintaining the balance between water and intracellular fluid levels. It maintains turgidity of cells. It is a process by which plants maintain their water content despite of constant water loss due to transpiration. This process controls the cell-to-cell diffusion of water. Higher osmotic pressure protects the plants from drought injury. Osmosis also helps in absorbing water from the intestines to the blood. Applications of osmosis. It assists plants in receiving water. It helps in the preservation of fruit and meat. It is used in kidney analysis. It can be reversed to remove salt and water impurities from water. Osmotic concentration of osmotic generation of power. Next is the active transport. Active transport depends on Active transport depends on integral membrane proteins that selectively bind a particular solute and move it across the membrane in a process driven by changes in the protein's conformation is the energy dependent. And move it across the membrane in a process driven by change in the protein's conformation and it's energy dependent. Active transport is the energy dependent and energy dependent carrier mediated uphill flow of metabolite against concentration of or electrochemical gradient and osmotic forces. It occurs with the help of a carrier protein and also with the input of free energy. Its energy source is ADP or ions which exhibit an electrochemical potential. It is a major mechanism by which a cell maintains a constant concentration of ions and molecules inside it. So when you just look at the structure, we can see active transport is happening with the help of ADP. And just look at the diagram. Uniport, symport, and antiport is there. Active transport is of three main kinds like uniport. It is the unidirectional transport of only one kind of molecule at a time through a membrane channel. Symport. It is the simultaneous unidirectional transport of different kinds of molecules. Next, antiport. It is the simultaneous transport of different kinds of molecules in opposite direction. Some are transported inward and the others are transported outward. Active transport is divided into two types according to the source of energy used to cause the transport: primary active transport and secondary active transport. Primary active transport, in the sense, it is the transport of molecules against a concentration gradient by the use of energy from ATP. Primary active transport is the transport of molecules against a concentration gradient by the use of energy from ATP. A single molecule is transported. Ions like sodium, potassium, calcium are transported across the cell membrane. Sodium, potassium pump, calcium pump in the muscles, and the proton pump in the stomach are examples. Transmembrane proteins are involved. In primary active transport, solute movement directly depends upon ATP hydrolysis. So what is happening in case of primary active transport? It is a transport of molecules against a concentration gradient by the use of energy from ATP. So a primary active transport, it is the transport of molecules against a concentration gradient by the use of energy from ATP. A single molecule is transported. Ions like sodium, potassium, calcium are transported across the cell membrane. Sodium, potassium pump, calcium pump in the muscle, and proton pump in the stomach are examples. Transmembrane proteins are involved. In primary active transport, solute movement directly depends upon ATP hydrolysis. It is also known as a direct active transport. Enzymes that take part in this type of transport are transmembrane ATPase. The most basic ATPase universal to all animal life is the sodium potassium pump, which helps to maintain the cell potential. Redox energy and photon energy are some other sources of energy for primary active transport. The use of redox energy for primary active transport is the mitochondrial electron transport chain that uses the reduction energy of NADH to move protons. The use of redox energy from primary active transport is the mitochondrial electron transport chain that uses the reduction energy of NADH to move photons across the inner mitochondrial membrane against the concentration gradient is an example where redox energy comes into play. Photosynthesis is an example of active transport using light energy. The proteins involved in this case use the energy of photons to create a photon gradient across the membrane and also to create reduction power in the form of NADPH. There are many types of active transporters like PATPase, sodium potassium pump, calcium pump, proton pump, FATPase, mitochondrial ATP synthase, chloroplast ATP synthase, VATPase like vacuolar ATPase, ABC ATP binding tape transporter. In plants, ABC transporters are found inside the cell and organelles layers, for example, the mitochondria, chloroplast, and plasma film. ABC transporters have a role in pathogen reaction and detoxification. Next is the sodium potassium pump. This is one of the best example of active transport in animal cells. This pump transports Na plus ions out of the cell and K plus ions into the cell. thus keeping the intracellular concentration of Na low compared to the outside, the intracellular concentration of K high. The pump is driven by the hydrolysis of ATP. It uses about 30% of energy available to any animal cell. The pump is transmembrane carrier protein made up of 4 subunits. This pump transports Na plus ions out of the cell and K plus ions into the cells, thus keeping the intracellular concentration of Na low compared to outside and the intracellular concentration of K high. This pump is driven by the hydrolysis of ATP. It uses about 30% of the energy available to any one animal cell. The pump is transmembrane carrier protein made up of 4 subunits. It has 3 binding sites for sodium ions and 2 potassium ions and a phosphorylation site to accept a phosphate from ATP. The pump is driven by the hydrolysis of ATP. It uses about 30% of the energy available to any one animal cell. The pump is a transmembrane carrier protein made up of 4 subunits. It has 3 binding sites for sodium ions and 2 potassium ions and a phosphorylation site to accept a phosphate from ATP. It maintains sodium-potassium concentration differences across the cell membrane, maintains volume of the cell. It is essential for oxygen utilization by the kidney. The sodium-potassium is an enzyme found in the membrane of all animal cells. It performs several functions of cell physiology. The sodium-potassium pump must break ATP down into ADP in order to pump sodium ions outside the cell, while it pumps 2 potassium ions into the cell. The ATP phosphorylates the membrane protein as it binds to the sodium and breaks down, and it dephosphorylates the protein as it binds with the potassium. Cellular respiration must occur to add the phosphate back to the ADP, thus restoring the ATP. So when we just look at this procedure or the steps of sodium potassium pump, extracellular fluid is there, Na plus high, K plus low there. But cytoplasm, Na minus lower, K plus high. Cytoplasmic Na plus binds to the sodium potassium pump. Then Na binding stimulates phosphorylation by ATP. Phosphorylation causes the protein to change its conformation, expelling Na to outside. Next, extracellular K plus binds to the protein, triggering release of the phosphate group. Next, loss of the phosphate restores the protein's original conformation. K plus released and the Na plus sites are receptive again. The cycle repeats. Na plus K plus ATPase. It is composed of at least 2 different membrane spanning subunits, a larger alpha subunit which carries out the transport activity and a smaller beta subunit which functions primarily in the maturation and assembly of. It is composed of at least 2 different membrane spanning subunits, a larger alpha subunit which carries out the transport activity and a smaller beta subunit which functions primarily in the maturation and assembly of the pump within the membrane. A third gamma subunit may also be present. The 2 rubidium ions are located where the potassium ions would normally bound. The ratio of the Na plus K plus pumped by the Na plus K plus ATPase is 3:2. For each ATP, the hydrolyzed 3 sodium ions are pumped in. It is an example of P-type ion pump. The P stands for phosphorylation, indicating that during the pumping cycle, the hydrolysis of ATP leads to the transfer and released phosphate group to an aspartic acid residue of the transport protein. Next, you can see the picture: extracellular region, plasma membrane, Na plus, cytosol, E1 conformation, then E2 conformation. Step 1. The pump in the E1 conformation and the ion binding sites are accessible to the inside of the cell. Protein has bound 3 Na plus and an ATP. A gate with the protein has been closed, shifting the pump into the occluded E1 state, in which the Na plus ions no longer flow back into the cytosol. Step 3. Hydrolysis of the bound ATP and release of ADP. That Step 3 to 4 causes the change from E1 to E2 conformation. Step 5, the binding site become accessible to the extracellular compartment and the protein loses its affinity for Na plus ions, which are then released outside the cell. The 3 Na plus ions are being released and the protein picks up into K plus ions. The closing of another gate within the protein shifts the pump into an occluded state and prevents the backflow of the K plus ions into the extracellular space. Step 6 to 7, occlusion is followed by dephosphorylation, and then ATP binds, which induces the return of the protein to the highest original E1 conformation. In this state, the binding site is open to the internal surface of the membrane and has lost its affinity for K plus ions. Functions of sodium potassium pump. The Na plus K plus ATPase helps to maintain resting potential, affects transport and regulates cellular volume. Sodium potassium pump helps to maintain the osmotic equilibrium at membrane potential in cells. It maintains the gradient of a high concentration of sodium excellularly and a higher volume of potassium intracellularly. Muscle contraction is dependent on a sodium potassium pump. The kidney have a high level of expression of the Na plus K plus ATPase with the distal convoluted tubule expressing up to 50 million pumps per cell. The sodium gradient is necessary for the kidney to filter waste products in the blood, absorb amino acids, reabsorb glucose, regulate the electrolyte levels in the blood, and to maintain pH. Sperm needs the Na plus K plus ATPase to regulate the membrane potential and ions, which are necessary for its motility and the motility of the acrosome functioning during penetration into the egg. Secondary active transport. The secondary active transport help in the movement of the ions with the help of the pore forming proteins. In this process, the channels are formed in the cell membrane by using electromagnetic gradient. The secondary active transport helps in the movement of other molecules against a concentration gradient. This gradient is known as cotransporters. Cotransporters are categorized into two types, symporters and antiporters. In case of symporters, the solute and the ions move in the same direction across the cell membrane. In antiporters, the ions and the solute move in the opposite direction. Two types of molecules are transported at once, like glucose sodium pump, Na plus Ca2 plus exchanger, and sodium or phosphate co-transporter are examples. Then co-transport. The movement of glucose across the apical plasma membrane of epithelial cells against the concentration gradient occurs by a co-transport with sodium ions. The transporter protein called the Na plus glucose co-transporter moves two sodium ions and one glucose molecule with each cycle. In the transport of glucose and sodium, the carrier molecule binds simultaneously with sodium and glucose at the outer surface of the plasma membrane and forms the carrier glucose Na plus complex. The complexing activity and the carrier induces conformational changes in it. As a result, the complex moves inward. On reacting the inner surface of membrane, the constituent of the complex dissociates from each other. The carrier returns to the outer surface for transporting another molecule of glucose. Carrier protein is there, Na plus glucose, exterior interior plasma membrane, then Na plus active transport of Na plus along with ATP. The transport cycle of a secondary transporter. The bacterial symporter Lutein family have 4 different conformational states during the transport cycle. Step 1, the outer gate in the protein is open, which allows both Na plus and leucine to reach their binding sites from the extracellular space. Step 2, the outer gate closes, occluding the substrates within the protein. Second leucine molecule bind to another site just outside the outer gate. Step 2, the outer gate closes, occluding the substrates within the protein. Second leucine molecule bind to another site just outside the outer gate. The inner gate opens and the substrates are released into the cytoplasm. The protein returns to its original state when the inner gate is closed and the outer gate is opened. When we look at the structure, extracellular region, outer gate is there, inner gate is there, and molecules are getting transported. Ion channel. Ion channels are pore forming membrane proteins that allow ions to pass through the channel pore. Ion channels are present in the membrane of all cells. The rate of an ion transport through the channel is very high. In many ion channels, passage through the pore is governed by a gate which may be opened or closed in response to chemical or electrical signal, temperature or mechanical force. Operation of cell in the nerve system, contraction of the heart and or skeletal muscle and secretion of the pancreas are examples of physiological process that require ion channels. Ion channels in the membrane of intracellular organelles are important for regulating cytoplasmic calcium concentration and acidification of specific subcellular compartments like lysosomes. Most ion channels are selective, allowing certain ions to pass through. Some channels conduct only one type of ion, like potassium, whereas other channels exhibit relative selectivity, for example, allowing positively charged cations to pass through while excluding negatively charged anions. Types of ion channels. Based on the stimulus to which they respond, ion channels are divided into three subfamilies like voltage gated, ligand gated, and mechanosensitive ion channels. Non-gated ion channels. Non-gated ion channels are always open. They simply allow ions to pass through the channel, also known as leakage channels. This means there is no gating mechanism and ions are free to flow through the channel along the concentration gradient. For example, K plus leak channels. Gated ion channels also there. An ion channel in a cell membrane that opens or closes in response to a stimulus such as a neurotransmitter or a change in pressure voltage. Gated ion channels are controlled by proteins. Chemically gated channels are located on the dendrites and cell body of the neuron. How do they work? Most ion channels are gated. That is, they open or close either spontaneously or in a response to a specific stimulus. That is why most ion channels are gated. That is why they open or close either spontaneously or in a response to a specific stimulus. Ligand gated ion channels, binding of a small molecule to the channel protein. Voltage gated channel, change in voltage. Most ion channels are selectively allowing only certain ions to pass through. Some channels conduct only one type of cell, for example, potassium. Voltage gated ion channel. Voltage gated ion channels are a class of transmembrane proteins that form ion channels that are activated by changes in the electrical membrane potential near the channel. Voltage gated ion channels are essential for the generation and propagation of active action potentials. They are voltage sensitive. Voltage gated ion channels are essential for the generation and propagation of action potentials. They are voltage sensitive, ion specific. They are specific to sodium, potassium, calcium chloride. Important for excitable cells like neuron. Role in regulation of depolarization and polarization of neuronal membrane during an action potential. They are distributed along the axon. How does voltage open channels? Voltage gated ion channels contain intrinsic voltage sensors. Ion channels typically are closed at the resting membrane potential but open upon membrane depolarization. These channels detect changes in the membrane potential across the membrane. The voltage sensor spans the membrane and is thus exposed to the electric field. Charge resides in the sensor move in response to changes in membrane potential and trigger conformational changes of the channel. Structure and function. Voltage dependent channels are made of three basic parts: voltage sensor, the powerful conducting pathway, and sensitivity filter. Functional architecture of a ion channel: transmembrane protein, sensitivity filter, voltage sensitivity gate. Voltage sensitivity domain, pore domain, sensitivity filter is there. Types of voltage gated ion channels based on the major permanent ion. Voltage gated sodium channels. These channels are responsible for the generation of action potentials of long duration. Voltage gated calcium channels, they regulate intracellular calcium concentrations, and these channels regulate the release of neurotransmitters at synapses. Voltage gated potassium channels. They constitute the largest and the most diverse class of voltage gated ion channels. They generate the resting potential. Next, voltage gated chloride channels. These channels are present in every type. Neurons are involved in the regulating excitability and cell volume. They are contributing to the resting potential, membrane resting potential. Ligand gated channels. Ligand gated ion channels, also commonly referred to as ionotropic receptors, are a group of transmembrane ion channel proteins which opens to allow ions such as Na+, Ca2+, Cl2+, Cl minus to pass through the membrane in response to the binding of a chemical messenger, that is a ligand, such as a neurotransmitter. The fundamental role of ion channel in the transmission of electric impulses. Ligand gated channels whose conformational state depends on the binding of a specific ligand. This group of channels open in response to specific ligand molecules binding to the extracellular domain of the receptor protein. Ligand binding causes a conformational change in the structure of the channel protein that ultimately leads to the opening of the channel gate and subsequent ion flux across the plasma membrane. Ligand gated ion channels: messenger, channel open, channel closed, receptor, ion, and then outside of the cell membrane, inside of the cell membrane. Voltage gated potassium channels, present in all animal cells. Voltage gated potassium channels are transmembrane channels specific for potassium and sensitive to voltage changes in the cell's membrane potential. Open and closed based on the voltage changes in the cell's membrane potential. During action potential, these channels open and allow positive flow of K+ ions from the cell. Potassium channels are one of the key components in generation and propagation of electrical impulses in nerve system. Regulation of apoptosis, cell growth and differentiation, release of neurotransmitters and hormones, and maintenance of cardiac activity. Action of KV channels. They can be present in three functional states: quiescence state, closed conformation, the channel does not conduct the ions in this state; activation state, depolarization of the membrane results in positive charge of its intracellular part, causing conformational rearrangement of KV channels and making an open conformation. Inactivated state, the membrane remains depolarized. The majority of KV channels switch to the inactivated non-conducting state. The structure of one subunit of eukaryotic voltage gated K+ channel. The KV channel of plants play an important role in salt and water balance and in regulation of cell volume. The KV channel of animals play a role in muscle and nerve function. Voltage-dependent potassium channels are homotetramers composed of 4 voltage sensors and 1 pore domain. Eukaryotic KV channel subunits contain 6 membrane-associated helices named S1 to X6. These 6 helices can be grouped into functionally distinct domains. Pore domain, it contains a selectivity filter that permits the selective passage of K+ ions. Voltage sensing domain, it consists of helices S1 to S4 that sees the voltage across the plasma membrane. The S4 helix that contains several positively charged amino acid residues spaced along the polypeptide chain, it acts as a key element of the voltage sensor. Outside of the cell, inside of the cell, voltage sensing domain, pore domain, etc. The voltage sensing domain is connected to the pore domain by a short linker helix. Under resting conditions, the negative potential across the membrane keeps the gate closed. A change in the potential to a more positive value exerts an electron force on the S4 helix. This force causes the transmembrane S4 helix to move in that its positively charged residues shift from a positive position where they were exposed to the cytoplasm to a new position where they can be exposed to the outside of the cell. The movement of the S4 helix in response to membrane depolarization initiates a series of conformation changes within the protein that opens the gate of the cytoplasmic end of the channel. Once opened, more than 10 millions of potassium ions can pass through the channel per second. After the channel is opened for a few milliseconds, the movement of K plus ions is automatically stopped by a process known as inactivation. The inactivation of the channel is accomplished by movement of a small inactivated peptide that dangles from the cytoplasmic portion of the protein. When one of these dangling peptides move up into the mouth of the pore, the passage of ions is blocked and the channel is inactivated. Outside of the cell can be seen, pore is there, central cavity is there, cell membrane can be seen outside and inside of the cell, lateral window, cytoplasmic domain and inactivation peptide.